Okay, all yours.
Thank you. Today I'll be talking about a quantum biological neural correlate of consciousness. Of course, the order is changed a little bit from the abstract. I'll talk about quantum biology and neural correlates of consciousness as an introduction, and then get into action selection, which I believe is a neural correlate of consciousness, and the catecholinergic neuron electron transport hypothesis, which is a quantum biological mechanism. Then I'll talk about ferritin and the associated tests for ferritin that show it's a quantum biological agent and part of the CNET hypothesis, dopamine metabolism, and iron, calcium, and neuronal signaling. Please feel free to ask questions as I go along. Don't save them for the end. So, what is quantum biology? It's biological processes that can't be explained without quantum mechanics, and as that is a subcategory of biophysics, but it's a relatively new field. There are a few things that have reached kind of the level of factual examples, although there's not 100% agreement that these involve quantum biological mechanisms, but photosynthesis and the mitochondrial electron transport chain are two. I won't be going into those in any detail, but I will note that ferritin and neural correlates of consciousness are not generally recognized as being examples. Neuroscientists typically say something like, well, you don't need quantum mechanics to explain how neurons function. I think that's generally true, except for one example. All right, and then consciousness, when we talk about consciousness, people often refer to other things or various aspects of it, like cognitive processing or awareness, but all of these things really add up to the human experience of consciousness. We are anthropocentric by design, and we don't know what it's like to be an animal or a digital data processor. So we project our human experience of consciousness onto those. But we really have to understand that mechanism, the physics of the human experience of consciousness, before we can start applying that to anything else. Any questions? So what do we know about the human experience of consciousness? Well, we know that there are neural correlates of consciousness. This was a homunculus developed by Walter Penfield in 1937. He was a neurosurgeon, and he showed that cortical columns are associated with specific sensory inputs. He also made one of these homunculi for the motor cortex, and these are neural correlates of consciousness. They're generally viewed as factual by neuroscientists. I note that the cortical columns, which are about in the gray matter area here, extend orthogonal to the surface. So if you're talking about electric fields being generated by them, first of all, it's an extremely complex electric field. What's usually measured is the field out here, not the field that's orthogonal to the columns. The columns oppose each other and they're orthogonal to each other and they twist around. So it's very unlikely that electric fields can integrate anything. I've never seen a rigorous analysis that addresses this concern. And when you look at the entire cortex, it gets even worse. And the same is generally true for microtubules or really anything else. The cortex is just not a good place to integrate anything. Now let's use an example for this presentation of smelling a fire and seeing a donut and hearing the phone, which is a different part of the brain. These are sensory inputs that evoke an action potentially. You know, you might want to get up and investigate the fire, eat the donut, answer the phone. But what in here does that? Nothing. This is not the action selection mechanism. Any questions?
Just a
So this is,
trivial thing. I don't see your cursor.
you don't see my cursor?
Yeah.
Oh, here. Now do you see it?
Yes.
Yes.
Okay.
Okay, good. I'm so sorry for that. But I don't think that changes too much. You all can see the labels. So this is a tractography of neural signals that go from the cortical columns down to the basal ganglia. The basal ganglia is generally recognized by neuroscientists as being where action selection occurs. Three neuroscientists, Gurney, Prescott, and Redgrave, have perhaps the most concise model for how all of these diverse signals get grouped for action selection. So the experience of smelling a fire, seeing a donut, hearing the phone involves signals all across the cortex. And this is just a sagittal view. So there's the coronal view, and it's very complicated. And all of these come down to the basal ganglia. and they work together to coordinate for choosing an action. So again, what integrates this? There's this concept in neuroscience called the global neuronal workspace. But if that workspace is the cortex, how are these connected? You have neurons here, you have neurons here. How do the ones here connect here? If it's electric fields, well, they don't interact at all. The ones here are orthogonal. So this is not a good place for that integration to occur. Microtubules have the same problem. There really isn't a good explanation, and neuroscientists tend to ignore that binding problem, as it's called, because it just doesn't make any sense. But there are some that say this is happening down here in the basal ganglia. It's not a majority view. Any questions on this? Okay, so the Gurney-Prescott-Redgrave model says that those diverse signals group dynamically into channels. So let's call this smelling the fire, seeing the donut, and hearing the phone. They come down to the basal ganglia, which includes the striatum, the subthalamic nucleus, and other components as channels, and one is selected. But what exactly the mechanism that's selecting that is not identified by the GPR model. They have some ideas, but they don't concisely identify how all of these different signals could block out the others. Any questions? This is a different view of the action selection mechanism. It shows the corticostriatal loops. The cortex provides inputs to the caudate nucleus and the putamen, which are the striatum, and then the other parts get involved with the direct and indirect pathway. So the direct pathway promotes action and is associated with selecting an action. The indirect pathway inhibits action, and when the direct pathway selects an action, it blocks out the other pathways to prevent seizures. And this is a very helpful mechanism, so that seizures are very rare in a healthy brain. The conventional view is that the substantia nigra pars compacta just sits out here and modulates the striatum. It doesn't receive cortical signals, and it's just viewed as modulatory. And this is textbook neuroscience. GPR has a first-in-time problem, though. And GPR is really the best, most concise or thorough model for action selection. If you have these distributed channels, GPR says, well, one of them will lock out the others when it hits this action level. And it's set for all of these neurons because they don't communicate with each other. But it's unlikely that these distributed channels would all hit the action level at the same time. You could have multiple channels hitting it, causing seizures. So this is not really a thorough model. But neuroscientists will say, well, we don't need quantum mechanics to explain this. We have an explanation. But it's not really a very good explanation. And that's why I think the SNC, the substantia nigra pars compacta, or SNC, is important. It's a very unusual set of neurons. There's about 10,000 of these. This is one drawn by camera lucida, starts at the cell body or soma, and then the axon comes and branches and branches and branches, and you have this extensive axon arbor. There are 1 million possible synapses, but only about 20% of them are active, and they provide dopamine to the striatum. So this one neuron can interact with a large number of striatal dendrites, and that's unusual. Any questions? When you compare it to a kind of a standard neuron, and there are hundreds of types of neurons, and they're not all standard, but this is what most neuroscientists or people would think of when you mention a neuron, is that you have dendrites that integrate synaptic signals. There's like a one-on-one synapse, and they all integrate at the axon hillock. When it hits a certain level called action potential, it causes a signal to propagate down the axon, and it generates signals down here at these synapses. This is again the conventional view of a neuron, and the SNC neurons are very different. They're also different at the synapse level, where they don't form one-on-one synapses. So this is a cortical afferent in the striatal dendrite, and the earlier models envisioned something like this. But what actually happens is the dopamine neurons don't have their own dedicated dendritic connection. They form three-way connections with the cortical afferents. And this is very unusual. Professor Schultz studied this extensively in the realm of predictive reward signaling. So when the cortical signals fire because a selected action had a better than expected result, that creates a learning process where these dopamine varicosities can become activated. And again, this is 25 years old, more almost 30 years old. It's in textbooks. It's the way that people who delve into dopamine neurons a little more than at the normal level would understand them to work as a predictive reward signal, but not for action selection. That's taught in textbooks. Any questions?
Chris, I have a question. Is that the same Wolfram who wrote Mathematica?
I doubt it.
Okay.
It's Wolfram Schultz. He was a neuroscientist. I don't think he was a mathematician, but he might have been. I don't know. So recently, Professor Pascal Kaiser at Harvard Medical School found that for movement control, dopamine modulates moment-by-moment activity in the striatum to mediate action selection. So he came to the opposite conclusion that Professor Schultz had. When you look at Schultz's data, I think that there's some of this data that's buried in there, but it's not common. It's only these very large dopamine neurons. Most dopamine neurons are much smaller, and they don't have this intricate mechanism. But for the large ones, the axon is releasing dopamine to mediate action selection, but these neurons don't receive the cortical signals. So what's causing this axon to fire in coordination with the cortical signals to select an action? Because it's not, the dopamine neurons don't cause action selection, they modulate it based on the afferents from the cortex too. And this was predicted actually in 2018, 3 years before this evidence was published by CNET. And when I predicted it, I was told it was wrong by neuroscientists. No, that's not the way it works. But in fact, some evidence came along after that, which is usually notable when you're talking about scientific predictions. Professor Kaiser wasn't done. He kept working, and he showed that the axon arbors have action potentials generated in them by postsynaptic stimulation. These flashes are action potentials being generated in the extensive axon arbor in a live slice. So this is over the striatum and the arbors. It's separated from the soma. So the dendritic signals and the soma are not involved in these action potentials being generated. They're being generated entirely by the interneurons. There's no cortical afferents in a live slice, but there are interneuron signals. Get back to here. Okay, any questions? And again, this was great data, in my opinion, because it confirmed a prediction made in 2018, 4 years before this was published, that these signals could result in action potentials being generated in those neurons. I just note briefly that the locus ceruleus is very much like the SNC. It is a catecholaminergic neuron nuclei. Those are neurons that generate dopamine and noradrenaline. Dopamine is a precursor to noradrenaline, so the processes that generate dopamine in the SNC are also used in the LC plus additional processes. It has the extensive axon arbors, except that they're distributed through the cortex. It has, it's part of the corticostriatal loops. It's in the brainstem next to the basal ganglia, and the SNC and LC communicate with each other. It also has these weird three-way synapses. So there's a lot of similarities in the structure and function of these neurons. Any question? That's why I call it the catecholaminergic neuron electron transport theory. Having two switches involved in the cognitive action selection process complicates the analysis, and it's not generally recognized or understood that way. Probably the only person talking about this. If you look at the evolutionary progress of the brain in mammals, the basal ganglia and brainstem have been well conserved. 500 million years, it's generally the same structure. The cortex started out very small and then grew and expanded with evolution and cognitive processing. So human cognitive processing is much better than rabbit or lizard cognitive processing. Here's the brainstem and the basal ganglia. Sorry for not using the cursor in the right spot. But anyway, I just note that. Any questions? So that leads to CNET. So CNET was formulated based on several things. One was the observation that ferritin is associated with electron tunneling, Ferritin I'll get to in a moment, but it tunnels over 12 nanometers, and I found that to be very important because most electron tunneling in proteins and such is over 2 nanometers. So that was a long distance. And then I looked at where it was in the brain, and I was looking for pigment protein complexes, because in quantum biology, pigment protein complexes are a recurring theme that shows up in quantum biological functions. And I found that there was this unusual pigment called neuromelanin in these axons, in these cell bodies. But the neuromelanin is what I focused on first, because it's like a quantum dot at 30 nanometers in diameter. But then I said, well, this is a great distance between these cell bodies, up to 10 microns, 20 microns, and that's not going to get the electrons over. And I looked closer, I found ferritin, and found that ferritin had much better quantum tunneling properties. So the hypothesis was, well, somehow all of this stuff works together to move electrons between the soma. And also would have to route, because otherwise the electrons would just go wherever they want. So there has to be some electromotive force, because electrons move by electromotive force. The electrons, I hypothesized, would come from the chemical and electrical environment in these neurons, but I didn't have a specific environment. The electron tunneling, I didn't really have a specific mechanism involved or in mind. I hypothesized one mechanism for the switching function, other than the tunneling, which was from tests. But anyway, it was just a hypothesis. This cortical signaling to the striatum was, again, based on an electromotive force being needed for this, and it was unheard of and unusual. But as you've seen, subsequent evidence showed that these predictions were correct. The fourth component was based on iron release contributing to calcium signaling, which was seen in other neurons, but not these neurons, and has not been associated with report selected action in these neurons. That still has to be established. So that's part of the hypothesis, but there's a basis for that. Any questions?
Yeah. I don't quite understand this sketch. You have this ferritin that seems to be something like 30 micrometers large.
Yes.
How am I to understand that?
That
Is
is
that a
question.
huge molecule, or what is that?
Yeah, so the ferritin is, I'll get to that in a moment, but it's about 12 nanometers in diameter, but it forms unusual structures in the neurons, and I'll have that evidence later on, but that's a very good question.
Because I mean, you're drawing, you have the axons. You said the separation of these separate cells is about 12 micrometers. Is that correct?
It ranges. Some are close, maybe sub-micron, but some are very far apart, 10 or 20 microns.
But how are they then connected by the ferritin? It's just a structure which ferritin creates. It's not individual ferritin molecules.
Right. Yeah, it's multiple ferritin molecules lined up to allow sequential tunneling. So
okay. Now
ferritin is 12 nanometers. For microns, you'd have to have thousands of ferritin particles lined up.
Yeah.
And again, this is very unusual, but it was a hypothesis at the time. There's evidence I'll get to.
Okay, thanks.
Thank you for the question. CNET solves that first in time problem because it's routing energy. And so, this is smelling the fire and seeing the donut. And then you have, hearing the phone and a bunch of other stuff going on. And the brain is doing cognitive processing on all these channels, but the electrons would route to the strongest channel and help it to reach action potential and help to prevent seizures. So this is communications mechanism between these neurons helps to avoid seizures. It helps to explain how actions are selected without having a fixed level for all neurons, how these neurons can communicate. And it's a quantum biological neural correlative consciousness. There's more to it, but again, I've talked to the GPR people and they didn't respond. They responded initially, and then I brought this up and they stopped responding. So it's hard to get word about this out. It's hard to get any feedback, but I haven't gotten any negative feedback. Nobody's poked holes in it yet. I certainly welcome any constructive criticism. Let's talk about ferritin, because ferritin is an unusual iron storage protein complex. It's not like most proteins. It forms from 24 subunits that self-assemble into a hollow sphere. They then receive ferrous oxide, Fe2 plus oxide, or ferrous iron. They receive ferrous iron, not oxide. The ferrous iron is water soluble in the cytoplasm, and it finds its way to these pores where it's moved into the core. It's oxidized to ferric iron and stored as an iron oxide nanoparticle. Ferritin was discovered in 37. So it's not even 100 years that we've known about it, but it's been around for billions of years in living creatures. And as a result, it's in almost every cell of every plant and animal. I mean, it's a very common iron storage protein. There's also some evidence when I started this in 2018 that it formed connecting structures, but it wasn't really recognized as that. It was just viewed as, oh, well, there's a lot of ferritin in these glial cells between these neurons. I wonder why. But nobody had a real good reason. Some people thought it was a cause of Parkinson's, and if we just could get rid of all that, unnecessary iron, we would cure Parkinson's, but that was not correct. Any questions? Ferritin is part of something called the labile iron pool, which is in cells, and it controls iron. Iron is extremely important for cells, but it's also damaging. It creates reactive oxygen species, which are damaging chemicals that can cause all sorts of havoc. So ferritin is tightly controlled by the labile iron pool. The labile iron pool is a bunch of different proteins that work together to control the level of free iron, labile meaning free. It's not really free, it's bound to glutathione, but even the glutathione levels are maintained. Ferritin just acts as a storage. It stores iron and it releases it in response to signals from the labile iron pool. It does not control the levels, it's controlled by the levels. So this is how ferritin is viewed by biologists. Any questions? It's also, interacts with the serum iron levels, and it's just a part of a very, very complex process. There's only about 4 grams of iron in the human body, so iron is, well conserved. It's, conserved in the conservational sense. Something that's not as well known by people other than people who study the electrochemistry of ferritin is that it can store electrons for hours. And this is first observed decades ago, and about 16 years ago, this study showed that when you load electrons into the core of the ferritin by using a UV light or an electron beam, that it can hold those electrons for a long period of time. And it does that by reducing the Fe3 plus iron to Fe2 plus iron. Now, UV light does this, and ferritin is actually present in skin and corneal cells, epithelial cells, and other cells that get exposed to UV light. And it's probably providing a protective mechanism. In fact, it's been called a protective mechanism. So it takes that UV light energy, an electron from the shell gets migrated to the core, and it's held there. But this is the same result with an electron beam. So just loading the core with an electron beam or UV light had the same result. The core stayed in its condition with the ferrous iron inside, the Fe2 plus iron, the water-soluble stuff. It was not coming out until a chelator was added. And then once the chelator was added, the chelator forces the iron out, and it takes a while, it takes about 15 minutes. But you had pretty much the same release characteristic. So this shows that ferritin is effectively a nanobattery. The labile iron pool provides better control of ferritin release, ferrous iron release from the ferritin than a chelator. A chelator just dumps everything out. The labile iron pool maintains a labile iron level. So in a cell, this could potentially last for days. Now, that's never been tested. And there are recycling procedures, but the interesting thing is even with the recycling procedures that cells use, the level of the electrons stored in the cell could remain the same for days. Any questions? Ferritin has also been shown very conclusively to support electron tunneling. This was an excellent study by Professor Christian Nijhuis in Singapore and his colleagues with a 1 volt potential across these electrodes. They measured a lot of data that was pretty conclusively showing electron tunneling through the ferritin at room temperature under ambient conditions. This hasn't been replicated in water, in solution of any sort, but the way this works, that shouldn't matter too much. Again, it needs to be done. I'm not aware of anyone who's done it. but it is proof that under electromotive force that would be seen in a cell under certain conditions, you could have electron tunneling from the core to something outside the core. Any questions? So this leads to the quantum biology of ferritin, which again, I might be the only person talking about this and publishing papers on it. But if the electrons are being stored in the ferritin, that could explain why ferritin behaves like an antioxidant. This has been observed by many biologists, and they can't really explain it, and why ferritin is overexpressed in response to inflammation, which is caused by reactive oxygen species and free radicals, or UV exposure in the skin, which in addition to UV also creates free radicals. So these two functions of ferritin are inconsistent with ferritin's place in the labile iron pool. It doesn't make any sense. People offer explanations like, oh, well, this could be related to iron in some manner, but it's not very rigorous. But if electrons are being stored in ferritin, ferritin can receive the electrons from antioxidants and buffer them, and then provide them to neutralize damaging free radicals. These two things would make a lot of sense. There's a bunch of other stuff. I won't get into it. I just note that there's also a lot of evidence that ferritin and melanin form combinations in various tissues and organs and cells. Melanin and ferritin are both have semiconductor-like properties. So it's very interesting, and I'd love to talk about it, but I'm going to not use up your time with that. How is electron tunneling determined? You can't directly observe it. You have to infer it from electron behavior that can't be explained classically, like ferritin storing electrons and neutralizing ROS and free radicals. But there are ways to test that more exactly so that you can get additional evidence of whether or not that's occurring. I've got some ideas to use quantum dot fluorophores or nitrogen vacancy nanodiamonds for that, but that hasn't been done yet. That limitation of quantum biological effects is generally true for everything. Entanglement, you can't see, you have to infer it from behavior. There's this idea that there's a collapse of the wave function, that the particle collapses to a point, but the wave function is a probability wave function. It's not a physical structural wave function. And the probability wave function has various physical attributes that are probabilistic. When you make a measurement, there's something called the quantum Zeno effect, where that measurement will persist for some period of time, maybe nanoseconds, but eventually the particle will return to its probability wave function, particularly in free space. When it's interacting with other particles, then it gets very complicated how the wave functions interact. Any questions? And this led me to develop a model for a bioelectric model for ferritin. And this is all based on a lot of work by others. The chiral structure and the electrostatic bias at the pores has been established, and it would explain how electrons could be attracted to the positive pores and the ferrous ions could be attracted to the negative pores, how electrons could get into the shell maybe from an antioxidant. When they're in, studies have shown that electrons move by polar ionic hopping through ferrohydrite nanoparticles. If there are two or more electrons in the core, they would repel each other and end up in a shell area between the nanoparticle and the protein. The reaction center where the ferrous iron is reduced to ferric iron is in the middle here, and the electrons would be generated, and somehow something happens to them. But then the ferric iron has to be reduced and it has to happen near a pore for the ferrous iron to release. I'm sorry, I'm walking all over ferric and ferrous. But in any event, this is my model. It's not terribly relevant, except I'm, I think, the first person to propose anything like this. It is relevant to this, though. It leads to this biomagnetic model because Professor Ron Nauman at the Weizmann Institute, who's somebody who discovered chiral-induced spin selectivity, a quantum biological and non-quantum biological physical mechanism, has shown that those chiral molecules can imprint ferromagnetism on otherwise superparamagnetic iron oxide nanoparticles. So ferromagnetic means it generates a magnetic field and maintains that magnetic field for some period of time. Superparamagnetic and paramagnetic means that it can be attracted by magnetic flux, but it doesn't have a remnant magnetic flux. It doesn't generate its own magnetic field. So ferritin is viewed as super paramagnetic. It's usually tested in bulk, where it would appear super paramagnetic. It's tested in environments where you don't have these transient magnetic fields being generated by iron storage and release and electrons moving in and out of the core. So I'm trying to do these tests too. If this model is accurate, it would explain why antioxidants donate electrons to the core. And this has been studied repeatedly. Vitamin C will cause ferritin to empty its core over time by loading it up with electrons that eventually cause the iron to be released. It takes a long time, relatively. The reactive oxygen species are paramagnetic, and they would be attracted to the core. And if the electrons can tunnel out, that would explain why ferritin behaves like an antioxidant. It's just buffering the electrons. So again, this is my model, and I'm trying to raise awareness of it, and I welcome any feedback if there's any questions.
Chris, is there a distribution of ferritin in the brain? In other words, is it localized more in some places than others, or?
Very good question, and that's my next slide, actually.
Oh, okay. Sorry.
So when I formulated this hypothesis in 2018, I didn't have great evidence of this. This paper came out about the same time, and I wish I'd had it, but it's an electron spectroscopic image of showing iron in red outside of the neuromelanin organelle. And the ferritin probably holds this iron because ferritin is what stores iron primarily. And it appears to be in layers where sequential tunneling could be occurring and where the electrons could interact with the neuromelanin particles. So this is inside of a neuron's cell body, a soma. Here are the somas over here in more recent data from 2025. And in between those soma are things called oligodendrocyte precursor cells, which have these processes that connect the cell bodies. And these processes are chock full of ferritin. The oligodendrocytes have the highest levels of ferritin probably in the body, but definitely in the glial cells in the brain, higher than neurons, higher than other glial cells. And what's weird about the oligodendrocyte precursor cells is that they usually mature in several weeks, and then the oligodendrocytes create myelin. Myelin is not created in the substantia nigra pars compacta. And these oligodendrocyte precursor cells don't mature. They're tissue resident for years, at least in this study in rats, probably in humans. But I could not have designed a better mechanism for this connection between these soma. And when I made this hypothesis in 2018, I did not know about these. So when I saw this paper late last year, I was very pleased to see that this structure was found by others, because it tends to validate the hypothesis at some level. I mean, there's no other reason for this to exist. I mean, it's very difficult to explain. It's very unusual. If you told a neuroscientist that they would find these, they probably would have scoffed and said, well, that doesn't make any sense. Why would an oligodendrocyte precursor cell be in neurons that don't have myelin? So any questions? But this does provide that structure, and I'll show you how that ferritin is located in those cells. I believe I've already gone over this. Yes, I have. So how do you test for electron tunneling? Like I said, you have to look for unusual electron behavior. And when I had this paper peer reviewed, one of the peer reviewers said, "How would you test for this?" And that's a very good peer review comment. So I added that, and I said, conductive atomic force microscopy might measure some unusual electron behavior. It hadn't been done before. It had been done on individual neurons, but not on tissues. And I didn't even know if it could be done, but that was one way that could possibly do it. So what is conductive atomic force microscopy? You have a conductive substrate, and you put the sample on top, and then a nanoscale cantilever bounces along the surface, and it measures currents in response to an applied voltage. It can also measure other stuff like height, material properties. And there's a lab, EAG Labs in Silicon Valley, and I reached out to them because I had nothing else to do after I published my paper, and I thought, well, why not? I'll check around and see how much that costs. And they could do it for a price I could afford. They said, send us the samples and we'll test them with conductive atomic force microscopy. And they're experts at it. So that was good. I was very pleased that they would do this. It took a little bit of persuading, but they agreed to. They tested similar materials, although they had never tested neural tissue before. So I called AmsBio because I didn't have any handy SNC tissue, and AmsBio is a tissue supply house. And I said, would you send some of these SNC samples to EAG Labs? They said, sure, pay us money and we'll put them on aluminum foil for you. This is a image of the tissue on glass. It's 5 microns thick. This is a 1 millimeter scale bar, so you can tell this is like a 1/2 inch square. It's very small nucleus. The SNC neurons are here. There's other types of nuclei around it. But in any event, this was sent with the glass tissue so we could index locations, and with the tissue on metal to EAG Labs. So if you can stain the tissue, this is what those cell bodies look like. And you can see that they're very close in places. They appear to be close. This is, like there's a 3-dimensional aspect to these cell bodies. So they're not really on top of each other like that. In 2 dimensions, in 3 dimensions, they're separated. But that separation distance can be microns. This is the tissue that was sent on the aluminum foil, and it's not stained, and it's hard to see, but fortunately the cell bodies stood out. The cell bodies are about 30 microns in diameter, and this is a 5 micron thick slice, so some of the slices cut through the cell bodies, and I didn't know if these were bare areas or what was there, but we said, let's test this. Let's also test some area that's out here, away from the high levels of neuromelanin and ferritin. There's ferritin and neuromelanin in lots of neurons. I won't say almost every neuron, but in a lot of neurons. But definitely ferritin. I'm not sure about neuromelanin. It's viewed as a waste product in most neurons. But we tested some of the control samples. So this is one control sample test, but it's 250,000 measurement points. And using a -10-volt bias on the probe, the electrons flowed to this conducting substrate with negative currents, as expected classically. Or if there was enough to insulate, the currents did not flow. So this was expected classical behavior. It was a good control sample test. It was the only control sample I did because I didn't have enough money to do this repeatedly, but I don't have any reason to suspect it wouldn't be the same. But when we tested the area around these big neurons, we had these really high positive currents, which should not be flowing, and they were dynamic. So this is a peak current. The test device measures 3 current levels, and this is the transient behavior. I won't go into great detail on this. And then we zoomed in on a high current spot. This is what it looks like from the side view. You can rotate the current data around in 3 dimensions. And we tested this area, and that area indicated that there was electron tunneling currents, positive currents, outside of these round structures that look like the size of neuromelanin particles in neuromelanin organelles. Now, why are positive currents indicative of tunneling? Well, the electrons are negative, the probe is negative, and electrons should not flow into a probe. But that's what those positive currents reflect, is electrons going into a negative biased probe. So this is, again, pretty good evidence. I mean, it's not conclusive evidence, and more testing needs to be done. I'm not aware of anyone doing this. I wish they would. They could test other tissues too. But the number of biologists who understand this is very, very small, vanishingly small. So semiconductor scientists understand this, and I convinced some to join me in a paper because of these tests and other tests, but in any event. So this lines up pretty well with the layers in this electron spectroscopic imaging. So these are 2 different test techniques, and they appear to have results that reinforce each other, which is, again, good evidence. It's not conclusive evidence. A lot more testing needs to be done. I mean, how do you prove something as a fact? You repeat the test over and over again in different labs with different equipment, you get the same results, and we're not there yet. So this answers one of the questions I believe Johan had about what is the structure of the ferritin in the cell. So in a cell, in an organelle called a lysosome, the ferritin forms these very dense structures. And again, these lysosomes are 3-dimensional, so there's one here in this slice, and there's others around it. There's some good evidence showing that these things connect up and can form a path. And the hypothesis is that electrons are tunneling sequentially through the ferritin. So that's kind of unusual thing. It hasn't been observed before. I think it's been observed here. Professor Kai Shen at the Ningbo Institute for Materials Technology and Engineering is a thin film nanostructure expert, and he agreed to do some tests for me to build semiconductor devices to test whether electrons are sequentially tunneling across between electrodes. This is height data from an atomic force microscopy scan, and you can see the ferritin from the height data is disordered. It has some order, but it's not really well ordered in this layer. But we used alternating layers to create order, higher order in one dimension. I'll show you that in the slide. But anyway, it's similar. We're modeling to some extent the ferritin in the cells to see if it has the long distance electron transport and the routing function. And I expected a different routing function, and the data didn't correlate to that, and I had to go back to the drawing board and figure out what was going on. But we used a 4-terminal device with gaps ranging from 20 to 80 microns to see just how far the electrons would move across a gap, if they were moving at all. To see if there was routing, we had the different electrodes that we could test. This is the idealized layer by layer process because the layers are disordered. They don't always line up in neat layer by layer structures like this, but in places they might. Again, these are nanostructures and they're very difficult to manipulate, but the layer by layer deposition process has been used for quantum dots in semiconductor devices and has been successfully used with appreciable yield rates. Now, the yield rate for these devices was only 25%. So 75% of these devices did not carry any more current with the ferritin than without. So the ferritin is not inherently conductive, but that's expected. That's the classical behavior. Ferritin has not been observed to be a conductor. What was unusual was when you had sufficient order, presumably, again, it would take a lot more analysis to determine what the order is. Quantifying order is not easy. I mean, how do you quantify order in 3 dimensions? I haven't seen a really good way of doing that in the abstract. If you can model the individual particles, it's possible. But again, we were looking for unusual electron behavior, and we found it. Any questions? So this is an example of one of the successful tests that had the unusual behavior. This looks like a diode type behavior where the electrons are flowing one way but not the other. It's a nonlinear behavior. This was a model with 20 micron gap and 3 layers of ferritin. It was retested, so we got these results. We took the dye out, put it in the refrigerator, came back the next day and tested it again, got the same results. This is in ambient room air. So this is very promising data to show that this long distance electron transport and a routing function is available. I mean, just going back to here, you have electrons going this way, but not going this way. And it looks like a diode, but that diode idea didn't make any sense as I was thinking about it. It's like, well, I looked at quantum dot tests and found that there's something called a Coulomb blockade that would explain this behavior. So in a Coulomb blockade, the electrons, if they're applied from a narrow end of a constriction, can go into the Fe3 plus core and reduce the Fe3 to Fe2. But then the electrons here can move to another Fe3 plus core without a whole lot of electromotive force being applied and can find their way to the other electrode. Going the other way, though, the electrons start to stack up. Electrons don't like each other and they repel each other and they form a Coulomb blockade. And you have to overcome it by increasing the voltage. We saw that in some samples. So, again, this is a reasonable inference of what's happening. Now, these electrons are strongly correlated. They're entangled. Their wave functions are entangled because they're bumping into each other. They're being forced to communicate with each other, and they can integrate information. So this integration mechanism, unlike microtubules or electric fields, has been shown experimentally. Now, it hasn't been shown to the point where we can say we integrated information from a bunch of different sources, but it's feasible. Any questions about this?
Is this Coulomb blockade important for you, this diode characteristic?
Yes, it is. It is very important to explain what's going on. The Coulomb.
So it's not only the conductivity; it must be directional conductivity.
Right. It's how electrons behave in quantum dot solids when they're tunneling around. So this Coulomb blockade behavior has been seen in quantum dot solids, where instead of ferritin particles, you have semiconductor crystals that are about the same size, 10 nanometers or less often. And sometimes they're put in materials that are kind of like neuromelanin. They're pi conjugated polymers. So it's very interesting that it's behaving consistently, in physical principle with other similar devices, other similar structures. It's a Coulomb well. The electrons get in, they're stored there, and they can't easily get out, but they could tunnel out theoretically. And this is pretty clear evidence of tunneling because they don't normally just flow.
Okay, but since I think the essential point for you is that there's many ferritin molecules connected together can link different cells.
Yes, and that the electrons can move between those cells.
Yes, and but why is it important that they should be able to flow in only one direction and not in the other? That's what I don't understand yet.
Because if they aren't being directed, then they're going to flow anywhere, and all those cells would be shorted out, and this would not work as a signaling mechanism. So a signaling mechanism has to have some sort of directional control, right?
Okay, now I get it. Yeah.
Yeah, I mean, this was very unusual. This hypothesis was just really, really, out there. I mean, most people probably viewed it as science fiction and not science, and rightly so, because it was just based on the barest of evidence. But the evidence obtained since then, like through these tests, has really emphasized it and supported it. So it does need to be retested. I'm not saying that this is the final word. I'm not out there preaching to audience that this is the answer to the hard problem of consciousness, but it could be part of it. So those are in semiconductor devices. So currently I'm funding live animal tests to test, anesthetized rats. So we can't test action selection, but we can test to see if this structure is present in the brain of a live rat. The team is doing that. Dr. Dwight Nelson is a former chief DBS research scientist at Medtronic, and he's advising. Professors Khalifa and Chatterjee are brain-computer interface specialists at the University of Florida, and they and their grad students are doing these tests. The tests are using a technique I developed. It's never been done by neuroscientists. They don't test neurons like this. It uses a high DI/dt pulse, which is very close to a Dirac function, which means that the frequency transform is broadband. If there are current paths at high frequencies available, then the currents will flow. And if there's insulators, then you won't see any flow. And high frequency response of neural tissue has been, pretty dismal. I mean, it's just the cortical tissue that's been tested doesn't respond to high frequencies. And most of the basal ganglia doesn't, but it's hard to tell because nobody tested at 20,000 hertz and above. They usually test down here. So this is in the cortex in multiple locations in one of the rats. We have this noise type signal, high frequency noise, but in general, the currents that are measured at high frequencies are very, very small. Any questions about this? This is the classical result. In the basal ganglia, when we got the probe into the SNC, and it wasn't easy, it took a lot of training to direct this probe to get it into the SNC. We use histology to confirm that. We had a high frequency time domain signal, and then we transform it to the frequency domain, and we can see that the primary components are 40 to 80,000 hertz, which is at a level that's much higher than any neuroscientist would expect. I mean, neuroscientists would believe that this is just noise. Hey, electrical engineer, filter that noise out. It's annoying, but I believe it's signal. And we had noise issues, and we solved the noise issues. So I don't think it's noise, but certainly until these tests are repeated at other labs by other people, we can't be certain that this is a signal in those tissues. And this is an anesthetized rat too, so it's a low-level signal. Sorry, any questions? So this kind of signal was seen at multiple locations in this second rat. And it had kind of the same profile. This was a third rat, and when the probe went into the SNC, And you can see the signals here are a little bit stronger here. I think this signal range comes and goes depending on location. I'd have to go back and look. But the point is that it's not just the same noise profile, it's a different signal profile. This is kind of similar. I mean, you could say, well, maybe that's a noise profile, but we don't know what the electron transport characteristics of those oligodendrocyte precursor cell processes are. And particularly when you stick a DBS type probe in here. But it looks like we're measuring a signal, and it's a signal that could possibly be modulated and used to test whether there's a reportable experience to the signal, if you could get it into a human, which is not going to happen for a very long time. But, you know, quite frankly, let me just bring this up. Deep brain stimulation does go into tissues that are connected to this. And it does change people's perception, conscious perception. So there may already be some evidence out there. I'm not going to get into that in this presentation. This was a big surprise. The zona incerta is not a catecholinergic neuron nuclei. It's kind of a connecting part of the brain, but it has a high level of ferritin. And it also had these high frequency currents. So it appears to be part of a pathway between the SNC. The SNC are in both hemispheres. And then the question is, well, if this is related to the conscious experience, why don't we have 2 conscious experiences or 4 with the LC? But this was very interesting data, which appears to show that the electrons can move beyond what was hypothesized in the CNET. Any questions? And the lateral hypothalamus, again, we were trying to get in the SNC, and these tests ended up there. The lateral hypothalamus has elevated ferritin. It's not as high as some of the other tissues, but it was apparently high enough to conduct these currents. Again, it's hard to say with just a small number of tests, but it looks good. So how do we know that there's ferritin in these tissues? Well, magnetic resonance imaging modulates magnetic properties. And even if this is a spion and not magnetic, it still acts as a contrast agent. People have used ferritin as a contrast agent by injecting it into the serum, the bloodstream. But this is endogenous ferritin in these tissues. And it's forming a contrast in the substantia nigra, which is one on the left, one on the right hemisphere. The red nucleus is another structure in between them. And then this study shows that the subthalamic nucleus and the zona incerta, which we tested, also have high levels of ferritin that act as an MRI contrast. The subthalamic nucleus is where deep brain stimulation usually targets. So there's evidence, again, that the probe in the STN is modulating people's experience of consciousness. There's a paper that says, I miss my old me. It's the results of somebody with deep brain stimulation. The deep brain stimulation is a technique, a therapeutic technique for people with Parkinson's. It helps them move. It helps them to overcome the symptoms of the disease. It helps them to have a longer life, but it is invasive brain surgery. And I just note that because ferritin is magnetic, we might be able to actually stimulate ferritin, this signaling pathway with high frequency magnetic fields using transcranial magnetic stimulation. I'm trying to get that funded. I have a team, Adam Khalifa, and then another professor at VCU who will do those tests if I can get money. But if you can get rid of the brain surgery for deep brain stimulation and just have the transcranial magnetic stimulation, that would be wonderful, because Parkinson's patients could get better treatment. So that's the first component of CNET. We've already seen the third. The second component was this idea that the chemical and electrical environment in the dopamine neurons could generate free electrons. In 2023, Professor Doug Brash at Yale found that the dopamine and noradrenaline metabolistic processes create chemi-excited electrons, excited triplet electron species. And a triplet electron is a high energy electron that's kind of stuck in a shell where it doesn't normally reside, and it can be damaging, and it's also long-lived. One of the questions from this is, well, if there are these chemi-excited dopamine and noradrenaline metabolites, why aren't they damaging these neurons? Something there must be neutralizing that energy. And they would be paramagnetic and attracted to ferritin, and they could provide an electron source for the signaling mechanism. So this is good evidence. It was in vitro, it's not in vivo. More work needs to be done, but it does come after the prediction that there might be something there, and it shows that there is something that could possibly be an electron source that's just there in normal practice. Any questions? And then the fourth component, again, I mentioned it was based on tests that had already been done. There's something called a ryanodine receptor in the endoplasmic reticulum. And Professor Hidalgo at the University of Chile and her colleagues have shown for the better part of 2 decades repeatedly that iron can stimulate ryanodine receptor-mediated calcium release. The iron interacts with hydrogen peroxide to create reactive oxygen species. And when the ryanodine receptors detect that, they release calcium. And that calcium release can induce a calcium release cascade called calcium-induced calcium relate, and it can contribute to calcium signaling in these neurons. So this has been tested in different neurons, not in substantia nigra pars compacta, but it's at least a mechanism that could explain how iron, so the electrons flood some of the neurons, they travel and they reach a level at where they force iron out of the ferritin. The iron interacts with hydrogen peroxide, and it causes this signaling mechanism to occur. So, again, when I started this, I was like, well, the electrons are moving. Now what do they do? Because electrons are negative, and you need positive stuff to make an action potential, because action potentials occur when the cell gets more positive and not more negative. And this explains how. And again, this was part of the reason why I went forward with the hypothesis, because I thought it seemed unusual that there should be this mechanism out there. It's not only in the neurons of interest, but in other neurons. It's also in muscle tissue. It's well established, but what's not in most tissue is iron. Iron is tightly controlled in the labile iron pool. So, this mechanism would not be used, in normal circumstances, and it wouldn't have been seen by neuroscientists or other biologists unless they understood this iron signaling mechanism. So that is the CNET hypothesis. If there's any questions, I'm happy to answer them. But let me just sum up and say, so the cortical signals come into the striatum. There's millions and millions of them. They're modulating the axons electrically. And that modulation effect can be integrated through the strongly correlated electrons that are bumping into each other in this ferritin electron transfer mechanism up here. So this could be what integrates. This could be the integrating function from those signals. And the global neuronal workspace is still there because the neurons are coming in from all over the neuronal workspace, but they're being integrated here for action. This mechanism doesn't work unless it's getting the signals. Without the signals, it's dormant and you would have an unconscious state. But with the right signals, you could have conscious state leading to action selection, or if everything's balanced, no action selection, free won't. So you don't have to act, but what was driving action is your neurons, not a little homunculus that's standing out here saying, hey, grab that donut, don't worry about that fire. So anyway.
I've got a couple of questions, Chris. Is there any thing that you can say about anesthetics and ferritin? Is there any